Good afternoon, thanks very much, and it's a great honour to be talking between the very smart guys from the HIPAA lab. The relationship between habitat stability and endemism has been found in a number of studies, and the basic principle is that as diversity accumulates over time, where the habitat suitability is continuous, that diversity can accumulate, and in other areas that diversity may be repeatedly lost, and the signal that you expect to get from that is one of endemism in stable areas. And a really nice example of this is a study by Anna Carnival and colleagues where they modelled the distribution of the Atlantic rainforest at 3 time periods going back to the last glacial maximum. And in the areas where the forest was predicted to be continuous, or the habitat was predicted to be continuously suitable for forest, they found within 3 frog species much greater genetic diversity in those stable areas and a signature of recent range expansion in the southern, less stable areas. So you're seeing that pattern of greater diversity in stable areas, and this level of looking at diversity within species is the right level to be looking for that kind of pattern if what you're interested in is Pleistocene climate cycles. Now, not every study has found that pattern. In fact, a study that we did last year, Carnival et al. 2014, found that stability didn't predict endemism in the southern parts of the Atlantic rainforest. So the system that I'm talking about today is a different rainforest system, the rainforests of Eastern Australia, which span over 2,500 kilometres down the east coast of Australia, and it's divided into a number of separate regions. And the connectivity between those regions has changed over time with the Pleistocene climate cycles. So we've got these separate areas, occasional periods of exchange between them, but it creates a great case study for this idea of stability and endemism. And there are 3 questions that I want to be able to answer. Firstly, where are the areas of interspecific endemism within this system? And secondly, how well can stability, current and past habitat, explain the locations of those areas? And as you'll see, to do this we actually need a better way of estimating the distribution of interspecific lineages, and so I'll be presenting that method to you. And the actual system within the rainforest that we're looking at are a number of species of frogs and lizards that are endemic to the rainforest. So they only occur in rainforest, and therefore the envelope of the rainforest also determines the maximum limits of their distribution. And what we're able to do was find that out of the almost 100 species that are restricted in this way to the rainforest, phylogeographic studies have been done for 53 of those species, and we were able to collect a range of published and unpublished data which identified evolutionarily distinct lineages, generally from mitochondrial DNA, with locations where each of those lineages has been recorded. So by identifying concentrations of lineage endemism, we're able to come up with the locations of our putative refugia where this diversity has been able to persist. Now, to give you an idea of the sort of data we're working with, this is a phylogeny. This is a phylogeny for the genus Saproscincus, and highlighted as one species, Saproscincus rosauer. And this one's actually been very well studied, and what you can see is each colour represents locations where specimens were collected, sequenced, and we know what lineage they're a member of. As you see, there are 6 different distinct lineages identified within that species. Now, if you want to study endemism, especially if you want to use rather than putting barriers on your map, you want to actually use a relative measure of endemism, you need to know range sizes or distributions. And so we need to extrapolate that from the points. Now, one way that's commonly done is simply by circumscribing the points with some sort of a polygon. And a problem with that method is that it puts a boundary at the very places we have least information, generally in a sampling gap, and you'll be putting arbitrary lines on your map, which you don't want to do because they're not based on the actual data. Another alternative is to use a species distribution model. Doing models for each individual lineage wouldn't be a good idea, because that then assumes that the reason a lineage occurs in one place and not another is because they're constrained to separate niches, and we don't have evidence of that either. So what we've done instead is Oh, by the way, the black dots are the points where we know the species occurs, but no lineage ID. And I should also mention that this is an uncommonly well-sampled species. I probably should have picked a poorer one to show how important it is to be able to intelligently interpolate into the gaps. But the method is the same. So what we do first is a species distribution model, and as you can see, the purple areas are the ones predicted to have the most suitable habitat. And then, while showing you the different steps, we start with the species locations and a set of relevant environment variables, and we fit a species distribution model. In this case, we use maxent, but a range of methods would do fine, as long as they'll give you a likelihood of occurrence between 0 and 1. Then what we do is we take the locations, of each lineage and apply a very simple principle, which is that if we're looking at a particular spot and we're wondering how likely it is that lineage A, B or C occurs there, it should be proportional to the distance from the known it should be inversely proportional to the distances from where we know what lineage occurs. But the distance we're interested in is not the linear distance, but actually the distance experienced by the species. And for that, we're using a cost-distance surface where the cost is habitat suitability. So what we're actually modelling is connectivity to known locations of the lineage through suitable habitat. And we come up with a cost-distance surface for each lineage, combine them using a weighting so that the models for the different lineages sum to the species distribution model. So all we've done is, in creating our separate models for each lineage, is to effectively partition the species distribution model based on accessibility to known locations of the lineages. So just to show you one more view of how this works, what I've done here is taken the same model and put a transect across it. And over here you can see the habitat suitability values from 0 up to a maximum of about 0.8. The bold black line along the edge is the species distribution model, and then that's simply partitioned up into the lineage models. So in a place like here where it's pretty certain what lineage occurs, you find that the lineage model is essentially the same as the species model. But in a place where you've got a gap in sampling or a number of close lineages, you'll find that for example here too, you'll find that the species distribution model is divided up amongst a number of lineages. And that's not indicating particularly co-occurrence, what it's indicating is uncertainty. And where you've got uncertainty, the best thing to do is actually carry that uncertainty through your model and maintain all the information you can. And that also flags places where additional sampling in the field may be most useful. So what we then want to do is take those 102 lineage distribution models and calculate our areas of endemism. And again, we don't want to put a threshold to divide our suitability values into presence and absence, because again, that's putting an arbitrary division into our data, which isn't actually based on, it's not adding information, it's actually subtracting information. So instead we use this model-weighted endemism function to simply work out what proportion of the prediction for each lineage is found in a given pixel, and we identify these areas, these concentrations of lineage endemism. And there's a latitudinal diversity gradient from north to south. But within that, we find very clear areas of endemism. And using a model method like this, we can be quite specific about where in the landscape that endemism is concentrated. Well, if we come up with a good prediction of it. And this is already useful in itself for example, for conservation, where we can help to direct attention to the areas where diversity is concentrated. But what we're interested in here principally is looking at how those areas of endemism relate to paleostability. And for this we use the method developed by Graham et al. Of modelling paleohabitat stability, where what you do is you take the current day distribution of the ecosystem you're interested in this case rainforest, and you generate a current day species distribution model, and then you project that back in time to a number of periods in the past. In this case, we had 62 time slices based on snapshot global circulation models going back 120,000 years ago, and we modelled rainforest for each of those periods. And then what the stability model does is it basically assesses the degree to which a particular pixel has been continuously suitable for rainforest over that time. It's a geometric mean of habitat suitability, and you can do that either statically, treating each pixel independently, or a dynamic model, as illustrated from that Graham et al. Study, where you're saying that the habitat can be continuous, which can be connected through space and time, even though it's shifted perhaps up or down slope or on latitudinal gradient, to track climate over time. And in our study, we did the static model and used the 10 metres per year as an estimate of the spread rate of rainforest, which was the middle rate that Graham et al. Used. Now, here is our stability model result, and the dark purple areas are the predicted areas of greatest stability. And okay, you can eyeball them and see that there's a broadly similar pattern. But before we infer any meaningful relationship between those two, we need to consider a number of alternative explanations. So it's possible, for example, that we simply have more endemic frogs and lizard lineages in areas that are better rainforest habitat in the present. So that would be our default explanation. Other possibilities are that areas of complex topography will have greater endemism. We know that that's a relationship that's frequently found. And of course we need to consider, if only to control, for the fact that smaller rainforest regions, such as here for example, or here, would have smaller distribution ranges and hence more endemism. So we compared all these possibilities using 7 predictor variables in a series of spatial autoregressive models. We compared all combinations of these variables, and using an AIC model selection framework. What I'm showing you here is the best model, which was found to be a very high likelihood, 97% likely, out of the range of predictors tested. And what we see is, yes, there's a role for region area, but we're really just controlling for that. And then even after we consider the current rainforest suitability, the strongest predictor is dynamic stability. So what we're saying is, if we want to explain where endemism is now, it's showing us clearly that we do need to clearly consider the habitat stability through the past. And another way of looking at that is with this boosted regression tree model. And we see 3 variables here. This is our dynamic stability. Here we have current rainforest suitability, and the y-axis is our lineage endemism measure. And what you see is that, kind of looking from front to back this way, you see that sure, areas of currently good rainforest habitat show a greater level of lineage endemism. But looking this way, you see this clear threshold here, and what that's telling you is that even if you've got excellent rainforest habitat now, you're not going to find a lot of lineage endemism if it gets below a threshold of about 0.45 in terms of historical habitat stability. So even where conditions are ideal now, low paleostability predicts low endemism. So the take-home messages are, as I said, you need to consider paleo's stability, and this effect is driven most strongly by the low stability end of the spectrum. I like to think of that as a filter where the low stability areas don't let the diversity through into the present. Oops. There. And we have this new method for modelling lineage ranges and also then calculating endemism, and that model's got a lot of applications for considering this diversity below species level in conservation planning or in a range of biogeographic and macroecological studies. I'm currently working on that with Craig Moritz and our group on looking at diversity across northern Australia, hopefully also applied in Brazil's Atlantic rainforest. And the details in the paper that came out in PLOS One in May, and the code to do all this will be online shortly. So thank you very much for your attention. I'd like to thank funders, my collaborators, and the generous providers of the phylogeographic data that we used for the study.